Hello again. So this is a pre-recorded lecture video, and this is the last module before your midterm. So I hope everyone is well prepared after this, and you will be able to study as well, because you can always go back to this YouTube video. And okay, so this is module 6 about biomolecules. And this is part 3 already. So first is we talk about biomolecules part 1, which is carbohydrates, protein, and lipids. And then part 2 is about vitamins and minerals. So this is enzymes and hormones, but I will be uploading a separate video for hormones. So in this video, in this lecture, we'll be discussing about enzymes only. Okay, so let's start. Okay, so first we have the enzymes. Let's define it. So enzymes are biological molecules. Typically, they are proteins, and they speed up the rate of chemical reactions that take place within the cell. So ang important word natin dito, key term or keywords are speeding up of reactions. Why? Because they are specific. Their structure is very important. So if you could see in this picture, so let me move my camera here. Okay, so if you could see in this picture in your right side, so that is an illustration. Parang Tetris, no? If you know the game Tetris, may mga blocks na bumabagsak, and then dun lang siya mag-fit sa certain block din. So ganun ang enzymes. They are very specific. We have this term or concept such as the lock and key mechanism or lock and key analogy. It was discovered by Emil Fischer. He was discussed in our first module in the history of biochemistry. So Emil Fischer was the one who discovered or somehow studied about enzymes, and he explained about lock and key analogy. So here, we all know na pag meron kang susi, it's only specific for a certain lock or a certain doorknob, no? Even a car, hindi siya magsisimula, or sa motor, it would not start without a certain or specific key. So we have the enzyme parts, no, here. Let me just, okay. So we have the enzyme parts here. So you can see the green shapes that we see here. Okay, this one. So that is our substrate. So the gray one that we see here is the enzyme itself. And where the substrate would attach is what we call the active site. Okay? So ayan yung ating parts. So I will be I hope you remember these terminologies because in repetition kasi tayo dito sa lecture na to. So, paulit-ulit kong sasabihin yung substrate, active site, enzyme. So, I hope na wag kayong malilito, no, with this. So, there are 4 steps in enzyme action. So, first is the enzyme and substrate. So, again, the gray globular protein here is the enzyme. So the enzyme and the substrate are in the same area. So that's our first step. And then second step is the enzyme. So grabs onto the substrate. So mag-a-attach yung substrate natin dun sa special area on which we call the active site. So yung combination ng dalawang yon, we call it enzyme substrate complex. So, yan na, no? So, nag-attach na si substrate sa active site. So, sometimes, the active site is especially shaped. So, area that fits around the substrate. And then, the third is when the catalysis happens. So, what does catalysis mean? So, catalysis is when the substrate is changed. So, it could be broken down or combined with another molecule to make something new. Or sometimes it will break or build chemical bonds. And when it is done, we call it the enzyme product complex. Enzyme products complex. So, just a review, no, dun sa first, second, and third step. Again, yung first step natin is yung substrate, and then the enzyme is in the same area as seen in the picture below. And the next picture is we have the attachment of the substrate to the active site, in which we call it now the enzyme substrate complex. And then third step is when catalysis happen, and now we have the enzyme product complex. And then the last step in enzyme action is when the enzyme releases the product. So this is the time when the enzyme lets go of the product itself and returns to its original shape. It is then ready for work on another molecule of substrate. So looking at the picture, ayan siya. So ito yung summary ng ating first step to last step. Okay. So first step is substrate and enzyme, same area. Second step is the formation of enzyme substrate complex because the substrate already attached to the active site. And then third step is when there is a catalysis and enzyme product complex is made. And then last step is when the products leave the active site of the enzyme. So enzyme in action, so we not only have the substrate and the active site and the enzyme itself, we also have other molecules that help in the shaping of the enzyme, no? So we have helper molecules such as cofactors. So it's a non-protein chemical compound, or usually it's a metallic ion that is required for an enzyme's activity to be a catalyst. So it helps in the biochemical transformation. So if you have seen the movie of Cinderella, one of the first Disney movie, and you know that yung slippers, no, yung shoes ni Cinderella is perfectly fit for her lang. So kahit sino pang babaeng magsuot nung shoes, walang nagkakasya kasi it was made only for Cinderella's feet. So parang ganun yung ginagawa ng cofactors, no. It helps the shoes, or it helps the enzyme itself to fit perfectly. So nag-a-add sa isang molecule or dun sa enzyme itself. So aside from cofactors, we also have coenzyme. So again, it's also a non-protein compound, but in this time, it is usually in the form of vitamins. So sometimes, often broadly called cofactors, no, but they are chemically different. Cofactor and coenzyme are different. So what's the difference between the two? So the cofactor, it is made up of metallic ions. For coenzyme, it is usually in the form of vitamins or vitamin products, no? So if you look at the illustration we have here, so this is from YouTube, no? It's a screenshot. So yung coenzyme natin is the yellow one here. So it will attach to the active site, and it cannot function alone because it requires the presence of an enzyme, no? But coenzymes, even cofactors, they attach temporarily to the enzyme body. So temporary lang siya. After the product leaving the site, no, it will be released na din in the environment around the cell. So dito walang forever sa cofactor and coenzyme. However, with the next helper molecule, we have the prosthetic group. So yung prosthetic group naman natin, they are partner molecules that bind tightly or covalently to the enzyme. So this time, itong prosthetic groups natin, they are permanently bonded with the protein. So one example of this is the heme. No, heme, we know that it is found in hemoglobin, myoglobin, and cytochrome. So sa prosthetic group, if you remember the difference between covalent and non-covalent bonds, sa covalent bonds, there is sharing of electrons, no, in an atom. And then sa non-covalent, there is an attraction between two molecules. So yun yung bond between two molecules. So dito mas mataas or mas malakas ang bonding if it is covalent, no? So kaya dito mas permanently bonded sa prosthetic groups. So, ayan, no? So, remember, we have 3 helper molecules. We have the cofactor, which is a non-protein, but a metallic ion, usually metallic ion. And then we have the coenzyme, which is also a non-protein, also a helper molecule, but it is usually in the form of vitamins. And then, again, it is temporary attachment only. And then we have the prosthetic groups, which are partner molecules that are covalently bonded and bonded with a protein. No, always with a protein. So if you look at the illustration we have here, so the illustration we have here, the red is represented by a prosthetic group. And then dito, parang ginagawa niyang perfect fitting yung ating substrate, no? Itong nasa gitna natin is the substrate. And then itong yellow part natin would be an coenzyme or a cofactor. And then now we have a complete enzyme and an active enzyme. So we have other terminologies that you should know, would be helpful. Okay, so we have an Apoenzyme, a Holoenzyme, and a holoprotein. Anong difference nung tatlo? So first, ang ating Apoenzyme, it's an inactive. Why? Bakit siya inactive? Because it lacks the coenzymes and cofactors. Wala siyang attachment at all. It's only an enzyme with an active site, but no substrate, no coenzymes, no cofactors. So again, kung makikita nyo, no, nakalagay dyan, inactivated enzyme. Okay, and then we have the next. We have the next here, which is a holoenzyme. It's an enzyme that is complete. So, kompleto with its coenzymes and cofactors as well as the substrate itself already attached to the active site. And then, the last terminology that you should know is a holoprotein. It is a protein with a prosthetic group or cofactor. So, dito naman sa ating holoprotein, there is only one present. It's either a prosthetic group, a cofactor, or maybe a coenzyme. So, ang analogy natin dito on how you would remember these 3 terms, sa Apoenzyme, no, think of it as an apo, diba? Like a grandchildren, no? Grandchild ay bata pa, no? So, hindi pa kumpleto yung features niya. So, now, wala siyang, diba, pag bata pa, hindi pa complete ang features. So, ganun din with an enzyme. It lacks the coenzymes and cofactors. And then, for holoenzyme, you can remember it by thinking of your lolo, which is, syempre, baka matanda na, when fully grown na, complete features, here, complete enzymes and cofactors. So, holoenzyme, lolo, no? Think of it as complete. And then, of course, the holoprotein, isa lang yung present. Liitan ko ng kaunti yung screen natin. Okay. There you go. Okay. So, now, we have this picture again. So, yung nasa gitnang picture is an Apoenzyme. So, Apo, bata pa. hindi pa complete, wala pa siyang cofactor, walang coenzyme. And then, you combine it with a coenzyme or a cofactor, which is the one on your right, this one. So, you combine these two together, and now we have a matured enzyme, which is a holoenzyme. Okay, so we have this term called Induced Fit Hypothesis. So this time it's when the active site undergoes a change. So nagbabago daw yung active site in its conformation, yung shape niya, to allow better fit between the active site and the substrate. And when an enzyme binds to the appropriate substrate, subtle changes in active site occur. So, if we look back to our steps sa ating enzyme action, again, yung first step natin. So, kailangan by heart, alam nyo na yung 4 steps, no? The first step is when the enzyme, yung bilog natin dito, and the substrate are in the same area. And now, if we look at it, no, yung shape ng ating first enzyme here is round. However, yung substrate natin on number 2 is triangular. So, here, nagbago yung shape ng ating active site in order for a better fit ng partner na substrate niya. No? So, second step is the enzyme substrate complex. And now, we were able to have a product, no, enzyme product complex, because nag-change nga, nagkaroon ng induced fit hypothesis on the second step. And now, there is a release of product, which is the last step in our enzyme action. The release of product, so now, bumalik sa shape ang active site. No, from here, umpisa niyang shape. No? So, that is induced fit hypothesis. Okay. Okay, so there are different conditions that affects the reaction rate of an enzyme. So, first is definitely temperature. There is a suitable temperature for enzymes to function according to studies, to my sources that I found. It is around 37 degrees Celsius. So, yun yung parang maximum na temperature wherein there is just the perfect timing, perfect condition for the enzymes itself. So, it affects the chemical bonding in active site. Thus, the rate of action is affected. So, in the next slide, so this is slide, you will see that, sabi dito, the decrease of temperature, magkakaroon din ng decrease of enzyme activity. So, looking at this y and x axis, in the y-axis, we have the rate of reaction, kung ganong kabilis yung reaction. And then, yung dito sa baba natin, on x-axis, we have the temperature in Celsius. So, kung mababa ang temperature natin, for example, here, we have 10 degrees Celsius. So, makikita nyo na yung rate of reaction niya is mababa. So, there is a decrease of enzyme activity. However, if there is an increase of temperature, more molecules collide. Increased chance that enzyme will collide its substrate. So, pag mataas yung temperature, yung enzymes natin, para silang malilikot, no? Para sila nagbabanggaan like this. And then, there is a chance, increased chance, na yung ating enzyme will collide to its substrate. And then, tendency, dahil nga masyadong mataas yung temperature niya, hindi mag-a-attach si substrate sa active site. And then, yung ating condition number 3, if there is a too high of temperature, it is possible, high chance that the enzyme will have denaturation, or there is a change of shape in enzyme. So, usually, mag-shrink siya, and it can no longer bind to a substrate. Okay, sorry, this should be substrate. Okay, it will no longer. So, sabi dito, enzyme denatures at high temperature, so rate falls rapidly. So, kita nyo naman dito na nag-increase na ng temperature above 37 degrees Celsius. So, bumaba yung rate of reaction kasi nagkaroon na siya ng denaturation. And then, we have another condition which is the pH, stands for potential hydrogen. So, potential hydrogen, it is basically the R scale of 0 to 14, which is acidity and basicity. Now, if there is an extremely high or extremely low pH values, there may be a complete loss of activity for most enzymes. Kasi we have an optimum pH for different enzymes. So, for example, we have here this table. So, lipase, which is found in the stomach. So, itong second part dito. Ang optimum pH niya is 4 to 5. So, if you know, di ba nga sa stomach, it's already acidic. So, yung activity ng enzyme dito is 4 to 5, which is acidic din. So, kung makikita nyo dito sa ating table here, illustration, sorry. Yung 4 to 5 is around here, which makes it acidic. No, and others, especially pepsin. No, pepsin is the one that breaks down protein. So, yan. Okay. So, dahil nga chances are maybe there is a speed of reaction, sometimes the enzymes need to be slowed down. Okay? So, meron tayong tinatawag na inhibition. When we say inhibition, it's basically restriction or parang hindering the process, stopping the process kasi nga, parang masyado nang mabilis yung reaction, ang dami na ng products na nagagawa ng enzyme. I think we should pause. Parang ganun sabi ng enzymes. So, if enzyme is making too much, there needs to be a way to reduce or stop a production. That is when inhibitor happens, no? Okay, so we have the first inhibitor, which is what we call competitive inhibitor. It's the molecule that blocks the active site. So, kung titingnan natin dito sa picture na to, ang inhibitor natin is the yellow one. The substrate is the red one. So, ang ginagawa ng inhibitor, totally, it will block the active site so that the substrate must compete with the inhibitor. So, meron ng complete, parang meron ng blockage dyan. No? So, wala na talagang makakapasok na substrate. So, tendency, the enzyme will be inactivated kasi nga walang substrate. Okay? So, next is we have a non-competitive inhibitor. So, dito, this inhibitor binds to the enzyme somewhere other than the active site. So, hindi siya sa active site na ga-attach, but to some other parts of the enzyme. So, kung titignan nyo dito, supposedly, di ba, pag competitive, ang binablock niya yung active site. So, dito, ibang diskarte ni non-competitive. Dun siya sa ibang part ng enzyme na ga-attach. So, ang tendency, mag-iiba ang shape ni active site. So, si substrate will no longer be able to attach kasi hindi sila perfect fit dun sa active site. Okay, so looking at the bigger picture. So, with enzyme A and enzyme B. So, again, ayan po yung picture natin with the competitive inhibition. We have the blockage of inhibitor sa active site. So, the substrate will now move towards somewhere else in the cell kasi hindi siya makapag-fit sa active site. And then, the one on your right, the purple one, is enzyme B, wherein it's a non-competitive inhibition kasi yung inhibitor nag-attach to some other part of the enzyme aside from the active site. So, the active site will change its shape and the substrate will no longer be able to attach to it. So, I will move on na kasi hindi naman natin, same lang to dun sa ating previous slide. So, we have another one, which is number 3, uncompetitive. So, dito, the inhibitor binds to the enzyme and substrate after they have bound to each other. And the products leave the site less easily. So, hindi ganun kadaling mag-leave yung ating product kasi meron ng blockage dito, yung purple one. And the reaction is now slowed down. No? So, dito sa uncompetitive, if you see, ito yung nasa third step ng enzyme action. If you remember, the third step is the enzyme product complex. Ito yung time na nakapag-attach na. So, nag-attach na si ating substrate sa ating active site. And it is now ready for release. However, dahil nga meron tayong inhibitor, uncompetitive inhibitor, may blockage dito, it will not be able to leave the enzyme. No? So, yun yung ginagawa ni number 3. Okay, and of course, we have the last type of inhibitor, which is an irreversible inhibitor. So, ang irreversible, ibig sabihin hindi mo na maibabalik, no? It's an enzyme that it's a molecule that permanently inactivates the enzyme. So, dito kung makikita nyo, total blockage na siya. And it will no longer be able to accept any substrate at all. Okay? So, to review, to summarize, we have the competitive inhibitor, which totally blocks the active site. So, a substrate will no longer be able to attach to the active site. And then, we have a non-competitive inhibitor wherein the inhibitor will attach to some part of the enzyme aside from the active site. So, nagbabago yung shape ni active site and the substrate will no longer be able to attach to it. And then, we have uncompetitive inhibitor. So, ang ginagawa nito, once nakapag-attach na si substrate sa enzyme, yung yellow natin, ibla-block niya yung enzyme and the product will not be able to leave the site. Okay? And of course, our last inhibitor, which is irreversible. So, wala siya dyan sa picture natin. But irreversible permanently inactivates the enzyme. Okay. So, that's the summary. Now, we have 7 major classification of enzymes. Pero dito, kasi you don't need to memorize this part. Because it's really easy to know, to figure out kung anong enzyme ang meron dito. So, based on the function. So, for example, we have oxidoreductases. Ang pinakang function niya is about oxidation reduction reactions. So, di ba, ang daling tandaan, oxidoreductases is for oxidation reduction. Transferases is for a group transfer. So, nagdadagdag lang tayo nung aces sa huli ng word. We have hydrolases. Hydro is water. So, anytime that you use water for a reaction to happen, you have hydrolases. And the next, we have lyases, which is the breaking of double bonds. Isomerases changes the geometry of structure. So, ang isomers natin, they are almost the same structure but with different atomic mass or molecule that is very different in some composition. No? So, yun yung mga isomers natin. And then we have ligases wherein there is a ligation and the formation of maybe a carbon-carbon or a carbon-sulfur, carbon-nitrogen or carbon-oxygen bonds dahil sa condensation reactions. And then of course, our last type of enzyme is a translocase. So from the word translocate, no, translocation, which means there may be a movement of ions or molecules across membranes or their separation within membranes. So, maybe there is a side 1 to side 2 translocation. So, yun yung purpose ng enzyme natin na translocase. Okay, so that will be the last part of our enzymes. And I will continue with hormones in our next pre-recorded lecture. And then after that, you will have your midterm examinations. So if you have questions about this Module 6 about enzymes, you may contact me through my email or through Messenger, direct message. And without further ado, I uploaded this slides in our Google Classroom. You can always check it out, always go back and review for hormones, and it will be an exciting module because you will be able to know which hormones increases when you're happy or when you're sad or when you're stressed, etc. Okay, so thank you for listening and happy studying.